In this section, we're going to discuss the differences in the types of chemicals that are involved in acid-base reactions and the different functional groups that are typically found in acid-base chemical reactions. So on the top, we've seen this a couple of times. These are carboxylic acids, and you can see the carbon double bonded to an oxygen, and that same carbon is bonded singly to an alcohol group. A common name for this example is acetic acid. So there are a number of different common carboxylic acid groups, and one is the most simple would be the formic acid, which is going to be the double bonded carbon to OH and a hydrogen here, that's formic acid. The IUPAC name is, this would be a methyl group, so methanoic acid. That's the IUPAC name. And some of these compounds, as this chain gets longer, we get to, they a lot of times have some less pleasant smelling odors. Some of those things in like Limburger cheese or some of those different kind of funky flavors or smell compounds tend to start to have a little longer chain. Once we get to perhaps 6 or 7 or 8, these become less volatile, so they are able to be aerosolized less easily, and then they become waxes and longer groups, and they have less of a kind of rancid smell. And so those are just some examples of the different carboxylic acid functional groups found in various compounds, biocompounds that you can read a little more about in your book. Your book discusses some various ways of naming these in terms of common naming versus IUPAC naming. I would only want you to know the IUPAC system of naming, but you can just read the book and see how there are other conventions for naming using Greek letters like alpha, beta, gamma, delta, for example, in terms of naming from this carbon to the longer the chain. So we start with alpha, beta, gamma as it gets longer and longer. But that becomes less useful as soon as we get past 4 or 5 carbons on that ring. So IUPAC naming, most of these have common names up to about 4 carbons, and then after that we typically get more use the IUPAC naming system, because then there are not common names for those longer compounds. Some of the physical properties associated with carboxylic acids are, we're going to look at this idea, if we have a carbonyl group, and then it's bonded to a chain, right? This is just the different number of carbons. We have 1, 2, 3, 4, 5, 6, right? As we get longer and longer, we're obviously going to see that melting and the boiling points are going to increase. So it takes more energy to break those apart. And part of that is, if we're looking at these compounds, we're going to have a partial positive charge on this carbon here, because electrons are flowing away from it to both of those oxygen. And so we'll have partial negative charges on these oxygens. And this we'll also have with this oxygen here and the hydrogen. I'll draw that out a little better for you. And we have this also partial positive charge because we have electrons flowing towards the oxygen. When we have this then, and we have other compounds in solution with that setup, right? We have this partial negative charge, partial positive, and there can be an interaction between these positives and negatives in the form of hydrogen bonding. And that hydrogen bonding is a strong intermolecular force, and that's what gives us a higher melting point. And as these longer R group chains are going to be able to also interact through those van der Waals forces, we're also going to be able to increase the temperature that's going to be required to pull these. The other thing we want to look at here is, as we get longer, and we'll notice that we have 1, 2, 3, 4, we get to 4 carbons. then these are all soluble. But then as we get into the 1, 2, 3, 4, 5, we have the 5 to 1 ratio here. We start to become less water soluble. So the solubility goes down as we get longer and longer. Remember that we talked about you want to have those 5 different hydrogen bonding capacity interactions for every carbon that is long, and that helps with solubility. Once we get above that, the solubility again decreases here. Also, as food scientists, you might recognize some of these: propanoic acid, butyric acid, valeric acid, caproic acid. Some of these start to provide some interesting flavors in certain cheeses. Small amounts are maybe okay, but if you get large amounts of butyric acid, that sometimes it's kind of described as a baby vomit flavor. So some of these get to be less appealing as the chains get longer, and then once they become so long, then again their boiling point or their aerosolizability is reduced, and then as these get longer, then they start to become less stinky because we can't perceive them with our noses, because they're maintained within that group and they're not in the air. Carboxylic acids like to undergo acid-base reactions, hence the name carboxylic acid. So if we draw this out and we have something like the sodium hydroxide here, and we know that sodium hydroxide falls apart into these different ions regularly, and we'll have these lone pairs of electrons on the oxygen will attack that proton, and then that will fall off. And in solution, we have this spectator ion that will associate then with our conjugate base here, which is going to be our carboxylic acid, and then the sodium is going to associate itself with that group now, and then we'll also generate our HOH, or our water, right? So these are the chemical reactions that you'll see here, kind of working through these different compounds. So there are different things we can generate depending on the starting materials. We might make water, and carbon dioxide, and these are common chemical reactions that carboxylic acids will undergo in the presence of various bases, and even organic bases, as we can see here and here. And then these are also, once this R group gets a little longer, these are also defined as different types of these are called carboxylic acid salts, and they're named in the same manner that we'd name inorganic salts. So here are some examples of the lithium comes first. So the cation comes first. That lithium is the name of that, and then it's the acetate. So that's the common name. Lithium ethanoate is the IUPAC name. Here we have the potassium is first, and then butyrate. So that's the common name for that carboxylic acid, but the IUPAC name is potassium butanoate. And then here is sodium benzoate. So the sodium comes first, and then that's a benzene. So this is actually a different group. That 6 carbons here is in a ring, and then we have this carbon coming off the side, O minus, and then you have the positively charged carbon. So that's a benzene ring will actually look like this. So that's a sodium benzoate. Once we get these become longer than maybe 5 or 6, these long chain carboxylic acids are actually called soaps. So if you have something a lot longer, and you'll have a carbon and the ions, the positive ion, this is kind of a soap group, and that's essentially what we're going to do when we'll talk about these a little bit more when we discuss lipids in the next chapters. The next functional group we're going to talk about is, we're going to kind of jump down to this ester group. And you'll notice that it looks very similar to the carboxylic acid, except for instead of this H, we have an R group. So there are different chains that can come off of this carbon here. And so that R group just is any number of our carbon or organic compounds coming off there. So here's an example of a methyl acetate. And the IUPAC name would be methyl ethanoate. So this is the ethyl group is the parent group. So that's the F here. And then we take off the E and add O8 on the end to tell us that it's an ester. And this group over here, depending on that group, right, that's a methyl group. It could be an ethyl, ethanoate, or a different chain group length here as well. So that's indicated in that prefix. Looking at some of the physical properties of these types of esters, you can see here we have the changing the length. This is the COOR group. So these are carboxylic acids, like they're drawn. This is the COO group, how these are represented here. And we can see a methyl formate, methyl acetate. These are the common names versus methanoate, ethanoate, and we're increasing the length here of the chain. Here we just have two different methyl versus a ethyl group on the other side of this ring over here. So these are some of the names, and then we will now look at some. Oh, here's also the ethyl benzenoate. So this is a benzene ring, an aromatic ring, and this off of here will have an ethyl group as well on the other side of that. Let's look at some of the physical properties now of these are some of the common names that were given before as well, and here are the different groups. You can see these are getting longer and longer. The molar mass is getting larger as we're increasing the number of carbons associated. And if we're looking at the boiling or the melting points, we see as we get those chains become longer, the boiling points will also go up. There are a lot I mentioned earlier that in the carboxylic acids, there are smells that are associated with those. And also for the esters, there are a lot of aromas that are involved in esters and what they smell like to us. Rum, for example, these are typically a little bit better smelling than the carboxylic acid versions of them. Here, this ethyl formate is a smell of a rum aroma. Methyl butyrate is a common apple smell flavor. Pineapple, pear, banana, jasmine, apricot, orange. These are all different flavors that are coming from these groups. You'll get a lot of, if we're talking about the brewing process and fermentation, a lot of these different flavors are developed and generated as a secondary metabolites from yeast generation, from yeast actually eating some of the sugars, and then these are some of the byproducts that are generated and created. So they can turn into really interesting flavor profiles out of those fermentation products. These are also compounds that these different elements or these different plants are making to generate these flavors if we eat them as well. And just so we can compare the difference between carboxylic acids and these esters, we'll notice that there are differences in their boiling points. So if we have formic acid and we have something that just has maybe 2 carbons in it, for example, 1 or 2, and even we go to acetic acid, the boiling point is 118 for the carboxylic acid versus the 54, 77, even if we get a lot longer. And remember that difference is this OH group, we can have hydrogen bonding. Where if we have the ester, we have lost the capacity to hydrogen bond because now we have this OC group, and no longer we can have a dipole, but we can have interactions in that way, but we cannot have hydrogen bonding. So this is a stronger intermolecular force, and that results in higher boiling and melting points. Carboxylic acids, we can turn them into an ester through this process called esterification, where we take a carboxylic acid plus an alcohol in the presence of a catalyst, an acid catalyst, and then we can generate this ester. And again, that's called esterification. Here's a specific example looking at acetic acid. If we take one butanol, so we have this methyl ethyl propyl butyl alcohol group, so one butanol and acetic acid in the presence of an acid catalyst, then we generate this butyl acetate. So you can see the green carbons come to this side, and we're going to generate this new compound. Really what's happening here is if we look at the chemistry that's happening, we have the lone pair. Here we have a partial positive charge, and this lone pair is going to attack that carbonyl carbon. And the bond here, what's actually going to happen is we're going to attack that, and we're going to generate this new compound. So we'll generate the water and then the ester compound as well. So this is the general kind of setup for the esterification reaction. This is also reversible. So this is an example of a hydrolysis where we start with an ester and we have water. Remember that last slide we just looked at, we generate water, and we can also go in the reverse reaction as well. So if we have hydrolysis, this is also called saponification, where we take an ester and we react with water, and then we generate a carboxylic acid. So we replace that OR group with an OH group, and then we generate our alcohol. So here's a specific example. If we have butyl acetate, we have this water, we'll generate that acetic acid and one butanol. Neither of these reactions will go to completion. They'll kind of have an equilibrium with each other. So if you only have butyl acetate and water, you'll start to generate some of these different compounds in your product. So there are different ways that we can manipulate this, right? We can add more reactants, so we can increase the amount of water, and maybe we can boil off our alcohol, for example, or get rid of some of these things in the reactant or the product to try to push this equilibrium one way or the other. If we're going the reverse reaction, if we're trying to generate the ester, maybe we would get rid of the water during that process to try to separate these out and drive the reaction to the right. A lot of times when we are looking at these types of chemical reactions, there are different ways of generating our product. So if we use something stronger than water as our base, remember I showed you that we're going to use those lone pairs to attack that carbon center. If we use something like hydroxide, sodium hydroxide, this is going to be a much stronger base, and we're going to be able to generate this. Now it's a carboxylate salt. So this is another way of doing this in a different way. We're generating a saponification reaction where we're making this salt instead of an OH group and an alcohol. And this is a lot of times what's happening in the manufacturing facilities for cleaning up fats on our equipment. When animals store fat, they store them as triacyl glycerols or tags, and this is the glycerol backbone. So glycerol looks something like that. It's an OH groups on each of these. So this is a glycerol back, and you can see right here, this glycerol backbone is right here. And here is our ester functional group. We have a COC or COR group, and here is our ester. So if we want to break this apart, if we add a base or an OH group, right? So this is negatively charged base group. We're going to be attacking this carbon center with our lone pairs, and we're going to break that apart, and then we're able to form, if we add a lot of the base, we're able to form our carboxylate salt. If we have Na or whatever, it doesn't really matter what that positive ion is. Those are going to associate here on the front, and this is going to be more like a soap. So we'll be able to clean this off a lot easier than this big functional group here. These are going to be, like we showed you, they're a lot more water soluble. They're a lot easier to clean off. So when we have big triacylglycerols, those get really sticky or kind of really slimy and get stuck on our metals and other things. They're hard to clean off. So if we can use these bases to break this apart and undergo this different type of esterification reaction, we're going to break those bigger chunks of carbon apart into smaller units that are going to have charged ends that are going to be more water soluble, so we can clean that off of our surfaces a lot easier. So this is the process that we're really going through when we're treating our equipment with bases to help clean them off, to get rid of those oils and those big long chains of carbon. I just want to also point out this analog, and it's discussed in your book as well, the phosphoric acid analog of a carboxylic acid. So we have this phosphate, and we have a double bonded oxygen. So that would be kind of essentially the similar to the carbonyl group, but it's a phosphate is the carbon instead of the carbon. And we have this OH group, so these alcohol groups coming off. And here on the left side is the OR group. So this is called an ester analog or a phosphoric acid. You can see we can do these phosphodiester, phosphotriester. These a lot of times we see these in biochemistry. We've looked at adenosine triphosphate, ATP. This is the kind of part of that backbone of that molecule that's important for harnessing different amounts of energy within these bonds and used for biochemical energy purposes. Now I'd like to go back up to the amine functional group that we've seen before and some of the characteristics of these. And you might recall that we have different ways of describing these. So just as an example, the difference between oxygen-containing compounds and nitrogen, where oxygen compounds are going to have these two different lone pairs associated with it. We're with the ammonia and the amines, we're only going to have one lone pair. So we have 3 bonds and 1 lone pair for each of these if they're in a neutral state or a non-charged state. And you can see that a primary amine is attached to 1 carbon, secondary amines attached to 2 carbon groups, and a tertiary amines attached to 3 carbon groups. You can actually have a quaternary amine, and that would be attached to 4 different groups, R, 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 and R. And that also means that this is going to be positively charged because we have to give one of these electrons up to another element, and then that provides a charge on that state. We'll talk about that, the importance of that, in just a moment. Here are a group of different types of compounds and how we go about naming them. Some of these compounds have common names, but the IUPAC naming system is usually to add this as a substituent group on that carbon. So we'll typically number the carbon starting at the nitrogen and go from there. So 1, 2, 3, 4, for example, and then it's a 1 amino. So we add that as a substituent group, as an amino substituent group. And so this would be aminobutane. Here is another example of a nitrogen attached within the middle of a chain. So we have 1, 2, and 3. We try to give that the lowest number possible in that instance. So it's at a 2. It's on the 2 carbon, 2 amino, 2 methyl. Here's the methyl group, methyl ethyl propane. So that's a 2 amino, 2 methyl propane. Here's another example where we're going to number it with the lowest number attached to the nitrogen. We have a methyl ethyl propyl parent group. So that's the propane. We have the number 1 methyl amino group. So we have on this is a amino group, and there's a methyl group attached to it. So that's the other way of doing that. You can also use this as a substituent. So N-methyl propanamide or propanamine. This would be another way to give that name. And then here is a dimethyl amino. So 2 methyls on the amino group, ethane. So this is the ethyl backbone. Below are some common names for different heterocyclic compounds, meaning that they are in cyclic form, but they're not necessarily, there are other compounds that are not carbon or hydrogen. And so here we have the nitrogen, a part of those cyclic compounds in some cases. And these are also known as alkaloids, meaning they have some basic function functionality to them. And since the nitrogen has the lone pair, they oftentimes are able to act as bases. So these are aniline, and some of the pyrimidines and pyridines, those are going to be imidazole. These are very common biocompounds or compounds found physiologically in plants and other cell types in biology. Here are some examples of some of the physical properties, and we can see that we have the boiling points are looking at 70, 80, up to 100, right? Because now we're able to add some hydrogen bonding to these different compounds, which give them an increased boiling point compared to their alkane counterparts. They're very miscible in water. When we get to be a bit longer, we'll notice that maybe we get a little less soluble in water, but typically we're able to get more of these in water than just the typical alkane. We have a comparison here between an alcohol and a amine functional group in terms of their boiling points, and you can see that they're similar in terms of their boiling points. Because of that capacity to hydrogen bond, these have typically higher boiling and melting points. Some chemical reactions, these again act like bases. So a lot of times these compounds in different types of environments, if you have different types of drugs, for example, that have amines on them, if we have HCl, you'll notice that the lone pair on the nitrogen can attack, and we are able to generate the HCl version of that. So sometimes we'll have this positive charge will help to make this more water soluble and this drug to be more easily taken up by our cells and get into solutions that we can take by a pill, for example. And so this is an example of if you see different types of drugs in the store that say something like, you'll notice that this compound is phenylephrine HCl is going to be shown in for example, you might notice that phenylephrine HCl, it has that HCl group in there, and that's really just, we know that there's a nitrogen in there that's probably taking on that positive charge to help it become more soluble. And finally, we're going to cover the amide functional groups here. And here's an example of a carbonyl group attached to an amine functional group. So these are one unit, and this is called an amide functional group. Here's an example of an acetamide. This is the common name. And if we're talking about IUPAC, it's ethanamide. So the ethyl group is the base compound, and then the amide is acting as the kind of the suffix for that, or that kind of functional group on the end is that amide functional group. Physical properties of the amides is they actually have really high boiling points and melting points for how the size of them. And this is good to look at a comparison with just carboxylic acids. So if we're looking at a formic acid versus formamide, where we're essentially substituting this hydrogen for an amine, we can all of a sudden, we almost double the boiling point. And because we're going to be increasing the ability for hydrogen bonding. So you can think about the nitrogen has hydrogens that can hydrogen bond to the oxygen of a carbonyl group of the same compound. So we can start to connect all of these together, where essentially each one of these molecules can have two hydrogen bondings, one associated with the hydrogen is attached, and then this oxygen can also hydrogen bond between another hydrogen and that other nitrogen. So we increase that amount of hydrogen bonding that can happen for these different kinds of compounds and greatly increase the boiling point for amide functional groups. Some of the different types of chemical reactions that we can undergo is we can interact an amine and a carboxylic acid to form an amide. So these are also sometimes referred to as carboxylic acid derivatives because they are derived from this functional group. So we've added these together, and this chemical reaction is really a substitution. We're substituting this hydrogen for a nitrogen group that's going to come in and replace that, and then we'll also form that water. So with that, that we're actually substituting the OH group completely for, we are substituting the nitrogen for this OH group. The alcohol group is going to come off, and we're going to create water and this carbon, this double bonded oxygen to nitrogen to the carbon. And so this is going to be the derivative that's formed when we increase these two, or put these two together. The reverse reaction is also possible. So if we have an amide functional group and we add water, and this typically, these are really stable, so this won't happen usually without the presence of some type of acid or enzyme catalyst that can help to break these bonds apart and then reform the carboxylic acid. So again, in biological systems where we have enzymes that can manage this and lower the energy involved, we can create this reaction to happen. Otherwise, this is more common, but this is also very slow unless you're heating this reaction up and perhaps moving some of the water out of the system to help drive the equilibrium. Amide functional groups, you can kind of see here, like C double bond oxygen to nitrogen, and it can be within a chain. It can be a secondary nitrogen, for example. It doesn't have to be a primary or tertiary. It can be any of those. But you can see the C double bond nitrogen, C double bond nitrogen. This is the backbone of proteins and peptides, and amino acids are going to have that carboxylic acid functional group, and then when they grow into this chain, they form the amide. So here is the kind of what this looks like, and we'll talk about this when we talk more about proteins, but this is an important structure in that backbone. So you can kind of see these proteins. It's the NCC, 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 where that every other carbon has a double bonded oxygen, and these between the nitrogen and one of the oxygens on an adjacent hydrogen, we can have these hydrogen bonds. So we can form really interesting structures based off of these secondary bonding structures, like these beta helices and alpha helices based off of that backbone hydrogen bonding. And again, we'll talk about that more when we talk about proteins in the next few chapters here. So this is all I wanted to cover for those different functional groups that especially interact in terms of acid and base reactions, and some of the most important functional groups for understanding some of the biomolecules we're going to see in the upcoming chapters here.